Welcome. Good afternoon, everyone. Let's get underway. I'd like to begin with prayer. Is there a volunteer to help us out? Please.
And Father, please help us that we'll be able to concentrate and focus on the material that's being presented so that we can learn and so that we can use this information to benefit our own lives and the lives of others. And we love the Father, see this in the name of thy Son, Jesus Christ.
Amen. Thank you. So I don't know that I think we are in the process of grading quiz 2. I think there's going to be a concerted effort to grade the exam tomorrow afternoon. With luck, most of that grading may be accomplished tomorrow afternoon, and so we'll see how it goes. But that's sort of where we are in terms of grading. Last class we had been discussing sort of the end of the chapter on carbohydrates, and we had been talking about some very specialized sugars that are termed glycosaminoglycans, GAGs. These specialized carbohydrates are formed of disaccharide repeats. And we had talked about one of these, hyaluronic acid or hyaluronate, and talked about sort of how it functions in the body and why its molecular structure and form allow it to have the properties that it does to accomplish its biological function. So we're going to sort of pick it up there and continue on. We will be using quite a few slides today. I apologize. I know that's a little bit less engaging than perhaps using the board or a mix, but I think for the sake of efficiency, we will, and also simply to allow for a clearer presentation of structure, we will use quite a few slides. So last class, we had talked about hyaluronate. You can, so it has these unique sugars. We have two glucoses that have been modified substantially. So let me just quickly refresh your memory. So over here on the right, let's see where we have this sugar that is glucose, but notice that its 2-hydroxyl group has been replaced by an amine. So that would be 2-glucosamine. That amine group has been further modified by acetylation. So this is N-acetylglucosamine. On the left, we have glucose, but we have a modification of the 6th carbon to this carboxylate. And consequently, this is gluconic acid, and this is coupled to the N-acetylglucosamine through this somewhat unique linkage of beta-1,3. Our bodies, our digestive tract, have enzymes that can hydrolyze alpha-1,4 linkages. This particular enzyme is called alpha amylase, and it is present in the saliva in small amounts, but it is present in the small intestine in larger amounts and allows us to break down things such as starch that we have in the diet. You'll notice that these particular linkages, the beta-1,3, the beta-1,4, would not be broken down by digestive enzymes that we have in our digestive tract. These particular sugars do attract a lot of water. I think I read somewhere that perhaps it will absorb 100 times its weight in water. That's really a massive amount of water that will be associated with these particular types of polymers. The consequence of this is they are extremely slimy, slippery types of gels, if you will, and that lends to their properties or their uses within the body. For example, we talked about the composition of synovial fluid in the joints is primarily hyaluronate. Because of its very slippery, slimy nature, it acts as a really remarkably good lubricant. Those negative charges with all the waters will slide over one another. Remember, there's electrostatic repulsion. Consequently, there's virtually no friction with the synovial fluid. It's able to keep things apart. We also talked about the hyaluronate being found in the orbit of the eye. It is the vitreous humor that is found in the interior of the eye itself, the orbit. And it has, again, this ability to be compressible, plastic, elastic, and so forth. But remarkably, it also does not absorb light in the visible range, allowing transmission of light through the entire orbit to the retina at the back to allow for vision. We talked about the hyaluronate being present in between cells, this idea of what's termed ground substance or the extracellular matrix. And you'll notice that if you compress polymers of hyaluronate, you'll squeeze out the water, and then you'll get to a point where you have electrostatic repulsion between these negatively charged gluconic acid groups. So it makes it very compressible. It acts then as a substantial cushion for tissues, preventing cells from rupturing, even with substantial physical force. You know, even if you didn't have this and were to clap your hands, you would rupture tens of thousands of cells, probably blood vessels, and it would be a substantial problem. Okay, so this is hyaluronate. We'll come back to it just a little bit. It participates in the formation of a sort of a super molecular structure called a proteoglycan. We'll see this in a minute. Any questions about hyaluronate? Yes?
Is this the structure we need to add now?
You should probably know what are the components of this. You don't have to draw this. More importantly, you should remember how it is used within the body and why it is useful, or what chemical characteristics that allow it to be useful in the way that it is. Yes?
Did you say that alpha amylase and other digestive enzymes don't break this down?
That's correct. And actually, it's kind of interesting. If you go to a health food store, even if you just go to your supermarket and go to the aisle with supplements, you will see bottles of hyaluronate and chondroitin sulfate being sold. The idea is that this is going to improve your joint health. The problem is that these cannot be absorbed. They cannot be broken down. Disaccharides cannot be absorbed. Remember lactose. If you don't have lactase, you are unable to utilize it, but the bacteria downstream in the large intestine have a field day with the lactose of milk and produce lots of gas and cramping, diarrhea, and so forth. So These supplements are extremely expensive, but probably completely ineffective because, one, they can't be absorbed. Secondly, there's really poor circulation to the joints. So even if you could get it into the circulation, the ability to actually deliver it to the space in the joint is probably not going to happen. Okay. Here's chondroitin sulfate. This, again, is one of these glycosaminoglycans. It is comprised of slightly different sugars. We still have the gluconate over here on the left, but now we have this sugar here, which is galactose. So galactose is the base sugar here, the basic sugar that has been modified. The 2-hydroxyl group has been replaced with an amine, so galactosamine, which has been further modified by acetylation to be N-acetylgalactosamine, but it is further modified with a sulfate. And these sulfates can appear on the 4-hydroxyl group, on the 6-hydroxyl group, and as you can see, these are going to be even more negatively charged, even more highly polar, and absorb probably even more water. The chondroitins are found, sulfates are typically found in cartilage and other connective tissue. It is thought that they allow these to be more pliable, more plastic and elastic. Again, you can bend these, but as you start to compress these strands in there, they will push back and give it this property of being able to reassume its original structure. So think of your ear or your nose. If you were to bend this or bump this, it doesn't snap off, it doesn't break and remain permanently broken unless you get hit really hard in the nose. But you have this ability to have a flexible structure in part of this, or maybe much of this is due to the presence of these chondroitins. Now there are other types of molecules, including collagen, that are present in these same tissues and contribute to their strength. Here, this probably contributes to their pliability, elasticity. Okay, so connective tissues then are rich in the chondroitins. They also participate in the formation of these superstructures, supramolecular structures, the proteoglycans. Keratin, again, it is found in a wide variety of sort of more solid structural types of tissues. You can see that it differs slightly. We have simply galactose over here, unmodified. But over here now we have the N-acetyl-glucosamine, and now we have sulfates on the 6 carbon of that. And so keratin, again, is found in connective tissue, cornea, fingernails, a few other structures that tend to be more, structural elements within the body. This particular glycosaminoglycan is heparin. Heparin is the most negatively charged biomolecule in nature. As you can see, both sugars have negative charges. In fact, they tend to have multiple negative charges. Here we have a carboxylate here. It can be either up or down. It can either be gluconic acid or iodo. I can't even remember the name of it, but it can be up or down as shown here. Here it's up, that makes it gluconic acid. This is modified now with a sulfate on the 2-hydroxyl group. Over here we have glucose, again with substitution of an amine at the 2 site But you can see that there are sulfates on multiple hydroxyl groups. The consequence of this, so this is found in mast cells. These are kind of part of the immune system. If you have an allergic reaction, the itchiness that you feel is often due to what allergies and histamines. Okay, so the histamines are released by these mast cells, causing itching and so forth. But the mast cells also release the heparin, and heparin, I don't know what it does locally there, but systemically it will bind to thrombin. Thrombin is a critical molecule in your coagulation cascade. And with binding of heparin to thrombin, you produce a state of anticoagulation. This is used clinically all the time. If you are going to operate on somebody, conduct a surgery, if you're going to have an indwelling line in their vein, you will continually wash with heparin to produce or to eliminate the formation of blood clots. Okay, so if you're cutting somebody, there is a natural process to produce clotting, and clots, if they move through the circulation, can lodge in the lungs, causing a pulmonary embolism, or they can lodge in the heart, causing a myocardial infarction or heart attack, often leading to death. So as a consequence, if they're actually deliberately cutting tissues and so forth, they will anticoagulate the patient and keep them on anticoagulation until they are confident that they are not going to produce blood clots. Again, if you have a line in your, if you have a needle in a vein and you're giving somebody an IV, you will flush that line, that needle, with heparin, just to prevent the formation of blood clots at the site where the needle is inserted. So this is heparin. Okay, any questions about this? Yes?
Alpha amylase can digest or cleave this.
It could potentially, if you were to eat heparin, but I've never, the only use I know is that it's actually infused into a patient. Okay, so they don't give them a pill of heparin. But presumably this could be. Now, as to whether it would be absorbed, I don't know. Again, given all of those negative charges, the ways of absorbing monosaccharides may not work for those. Yes?
Does it interact with thrombin through ionic interactions?
Yeah, just through electrostatic interaction. Thrombin has multiple positive charges on it. And so it simply binds it and effectively inactivates it by simply tying it up. All right. Now let me move on and we'll talk about one more thing, well, two more things. So this shows you, it's a little bit complicated looking, but this shows you one of these proteoglycans. And it is a unique structure. The core, this sort of, it's going to be hard to see this. Let me get something that's a little bit more easily seen. So the core of this is heparin. So we have this long polymer of these disaccharide subunits of, did I say heparin? It should be hyaluronic acid. So the hyaluronate forms the core, this long polymer of this disaccharide unit. Attached to that are proteins. So all of these little arms sticking out from there are proteins. And those proteins in turn are modified with chondroitin and keratin. And that's shown in the blow up here. You can see that close to the hyaluronate, we have keratin, and then in the red here we have the chondroitin. this forms these massive bottle brush-like arrangements, and these are present now in the intermembrane space, the space between cells. And they form this meshwork. They just simply remember now that all of these things are negatively charged, so all of those little tendrils, all of those little filaments sticking off are going to position themselves as far away from one another as is possible because of electrostatic repulsion. This ability to spread out allows other molecules to become tangled with the proteoglycan. So things like collagen or fibronectin, other fibrous proteins that are largely hydrophobic and fibrous in nature will be intercalated with the proteoglycan and form really a series of tangles. And these tangles really are holding the cells together. These are not tightly positioned one next to another. They're really this kind of open meshwork. Some of these are large enough to allow other cells to move in, but certainly they allow proteins to move easily through the interstitial space to cells within a tissue. You'll notice here that on the surface of the cell, there are these cell surface proteins called integrins. They will bind to the fibronectin. The fibronectin in turn gets entangled with the proteoglycan, which is in turn tangled with the collagen. And this is really what holds cells together. There are not covalent bonds coupling one cell to another. There's simply this meshwork of fibrous proteins and proteoglycans that are accomplishing that purpose. Suffice it to say that this acts as a cushion. As you squeeze cells together, the water can be displaced to the side, away from the pressure point. As soon as you let go or remove the pressure, then the water will come back in and fill the space. So again, this is how we can have a cushion such that typical wear and tear and running and pounding those feet on the pavement is not going to lead to massive cell death and bleeding in your feet. Okay, so we're blessed to have this kind of cushion. Okay, any last questions on glycosaminoglycans and their functions? Yes?
So when the fibrous proteins intertwine, is that done with electrostatic interaction as well?
No, they're largely hydrophobic. They're not going to be attracted to these proteoglycans. They simply can worm their way through there. They're not repulsed. Remember that that's a misconception. Water is not repulsed by hydrophobic compounds. But we do have a hydration cage that wants to be as small as possible. So, other questions? All right, and then we're going to shift gears and we're going to begin talking about chapter 8, which deals with nucleic acids. And we are only going to talk largely just about their structures. We're not going to really talk about their functions. That would be a full semester course. Chem 482 talks about RNA and DNA and cell division and the regulation of these processes. We will only touch on the structures of DNA, RNA, and some modifications of unwanted modifications of structure of DNA, which we'll call mutations. So this slide shows you a nucleotide. Up here, oops, we have this nitrogen heterocycle. Some of these are fused ring systems, 2 rings fused together. Others are single rings, but they're called bases because of the presence of these nitrogens with their lone pairs of electrons. We have this sugar down here. This is the sugar down here is ribose. In this case, we have deoxyribose, meaning that the 2-hydroxyl group has been eliminated. So this is the type of ribose found in DNA. RNA has the 2-hydroxyl group present. And then the final piece is this phosphate that is present on the 5-hydroxyl group of the ribose. These bases, as mentioned, come in 2 varieties. Well, actually come in 5 varieties. There are 5 different bases. 4 will participate in DNA, 4 will participate in RNA. These are the purines, as shown here. These have these 2 rings that are fused together. You'll notice that in this case, we have a free, sorry, it's a little hard to see the color. We have this free amine up here. This can hydrogen bond and will as we form double-stranded DNA. We have a free amine out here, and we have this carbonyl. This is a tertiary amine, secondary amine. Actually, I guess actually it's a tertiary amine. It's not very basic. Again, this is not very basic. They can hydrogen bond some, but not as extensively as a primary amine. These are the pyrimidines. These are single ring structures, but they're heterocycles. They have nitrogen in the ring. And externally, they have sometimes a primary amine or carbonyl that can hydrogen bond. This one has 2 carbonyls that would participate in hydrogen bonds as we see here as well. Okay, you will not need to draw these structures, but you should know that which are pyrimidines and which are purines. The organization of these nucleic acids is to form long polymers. This is accomplished by coupling a 5-phosphate group to a 3-hydroxyl group. So here we have the we have the 5-hydroxyl group here coupled to the 3-hydroxyl group here through this phosphate. So this is a phosphodiester bond. There are really sort of 2 ester linkages, one to each of the riboses in the chain. The phosphates with their charges are going to be oriented towards the water. This is going to orient the bases toward the interior because they are largely hydrophobic, though they have some polar characters or polar groups that can be present. The bases can couple. They can form these hydrogen bonding structures. Remember also that these bases can be stacked. Their hydrophobic nature or having hydrophobic characteristics allow them also or make it favorable to orient them towards one another and push them towards one another as you have them surrounded with water. A will couple to T, G will couple with C, and this is shown over here. Here we have the A-T bonding. In this case, we have simply two hydrogen bonding interactions. A carbonyl here will hydrogen bond with the primary amine. We also have this ring nitrogen coupling with a hydrogen over here. This is going to be a weaker type of hydrogen bonding because this is not neither of them is a particularly good base or acid. In the case of the C-G coupling, so G-C, we have 3 hydrogen bonds. And again, you can see that some of these are going to be stronger in nature. So having a carbonyl interact with a primary amine, as we have top and bottom here, will produce stronger hydrogen bonds than these ring nitrogens interacting. But nevertheless, what this means is the more GC character you have, the more energy is required to cause double-stranded DNA to dissociate into single-stranded DNA. This is called melting. So when somebody in the laboratory talks about melting DNA, they're talking about adding enough thermal energy that you're able to part the two strands, the two single strands of DNA from double-stranded DNA. Okay, as you form double-stranded DNA, it requires that you have complementary bases all along. That is to say that you don't have the occasional GC coupling, but you really need GC-AT coupling really over almost the entire structure of the double-stranded DNA. As this forms these base pairs, it creates a structure that is shown here. This was discovered in part by Watson and Crick back in the day, for which they received the Nobel Prize. But the structure this is also called the B form of DNA, and it has a major groove. That is, we have this larger opening here and down here. Can't really see it. And then we have this minor groove that is present in between the major grooves. Major in the sense that it is more spacious, it is a wider opening, and the minor grooves tend to be fairly narrow and less accommodating for insertion of other molecules. The major groove is used in some instances to allow for the binding of proteins. These proteins then will help regulate the transcription of the DNA, or they will inhibit transcription of DNA. So these are called transcription factors, these proteins that can interact with the double-stranded DNA. Okay, there is a predictable series of dimensions for these. These are summarized in a chart up here. I'll show you this one. So the B form, the central column, is what you should know best of all, because this is what DNA, the structure that DNA assumes naturally. You can see here there's also an A form. It is an experimental artifact. That is to say, you can produce this if you crystallize double-stranded DNA from a non-aqueous solution in the lab. It's not something that occurs naturally in cells, in organisms. The Z form happens when there is an extended range of GC, just GC base pairing. So it requires no AT, it requires an extended array of GC pairs. It is its function is not fully understood. In most cases, it seems to be associated, when it's seen, it seems to be associated with diseases. Whether it does, I think, have, or at least it's been proposed to have some regulatory role for interferon, but this is beyond really what we need to worry about in the class. Suffice it to say that it can be seen, but in nature, but primarily seems to be associated with problems, health problems, diseases. All right, questions about DNA structure? Yes?
What's the main function of the minor group? Does it also operate as like a site where the protein are going to come in, either inhibit or promote? Or what's the main function of the minor group?
I think there are transcription factors that do interact with the minor groove, but just based on the space available, most of the interactions with proteins involve the major groove. I'm not sure that it has, if you will, a significant independent function. It's primarily in conjunction with things binding to the major groove. Yes?
You also want to know the dimensions of DNA, the angstroms.
I would know the dimensions. I don't think you need to know the last little bit down there. So the last two things, the sugar pucker or the glycosyl glycolytic bond orientation. Okay, we're going to talk now. That's it for DNA. Thank you. And we're going to move on now and talk about RNA. RNA does not. So for DNA, you have two independent copies of DNA, and they form these double-stranded structures. Those are the important biological structures for DNA, the double-stranded. RNA, on the other hand, is a polymer of nucleic acids, but it is a single molecule. If you have, as shown here, if you have RNA and it has just an extended open form as displayed here, it actually assumes a structure that would be equivalent to the A structure of DNA in terms of its dimensions, it being a right-handed spiral. It is obviously not DNA, and it doesn't form double strands, but it does tend to adopt dimensions and a general overall three-dimensional structure that is similar to the A form of double-stranded DNA. Here are some secondary structures of RNA. And you can see, what you see here is that we can get base pairing for RNA, but it is within the same molecule. So rather than bringing two copies of DNA together and forming this double-stranded DNA, we'll see here that there are regions. So let's say right here, we can see that there are regions where the RNA molecule will double back on itself and will base pair. You can see that there can be incongruities in the base pairing. And this can lead to, in this case, a bulge. So let's just say a single nucleotide that has no partner. Or if we have multiple in a row, it can lead to this loop produced internally. And we also see this other kind of interesting structure where there is there is base pairing, but it leaves the loop at the end, and this is called a hairpin. Hairpin turns in RNA are very important. They, in fact, all of these elements give rise to a particular three-dimensional structure for the RNA. RNA is very interesting. So some RNA is messenger RNA, and this will carry the genetic code from a gene to the ribosome. There in the ribosome, the genetic code is translated into the addition of individual amino acids to a growing peptide polymer, a protein. But other types of RNA have very particular structures. For example, transfer RNA is also the RNA that brings specific amino acids one at a time to the ribosome and allows for them to interact with the ribosome and transfer the amino acid from the transfer RNA to the growing peptide. So they have reproducible three-dimensional structures. So in effect, they have a native conformation similar to proteins. If the transfer RNA or the ribosomal RNA is to function normally, it has to have the same structure. In the ribosome, it gets really messy because there are, I think, as many as 50 different proteins, and there are ribosomal RNAs, and they all have to bind together. And these RNAs have particular structures and functions. They have to bind to the same protein or the same ribosomal RNA in the same way time after time after time, or you lose function. So their three-dimensional structures are as specific as those found for proteins. It allows them to interact with specific other ribosomal RNAs or proteins present in the ribosome. So here just gives you a sense of what the structure now of a hairpin turn is. You can see that it does have a very distinctive look to it. Some of these structures, so I think this might be a transfer RNA here. I'm not absolutely sure, but let's guess that this is a ribosomal RNA. And these require then the same reproducible final conformation time after time. Okay, we're going to spend, and again, you can get these interactions between the ribosomal RNA and a peptide transcription factor. So there is recognition between these molecules, and they can be regulated, their function can be regulated as suggested by this slide. Okay, we're going to talk about mutations very briefly. These are summarized sufficiently in the text. They're summarized in the slide. I'd simply like you to know the types of mutations that can occur. Again, you don't have to draw structures for these. One type, I don't know how common a type, but one type of mutation is the formation of these thymine bridges. So if we have one on top of the other two thymines with UV light or other ionizing radiation, they can form these peculiar covalent bonds. So we're talking about now free radical chemistry. The ionizing radiation produces a free radical, which is highly reactive and it's stacked right on top of another thymine. This can lead to the cyclobutane form of things down here, or a single bridge between the thymines. This is problematic, okay, because we are now modifying its ability to probably hydrogen bond effectively. In some ways, we're changing the ring structure. So these become, these then, if we're talking about replication of DNA, this would pose a problem. You can have deamination. So these extracyclic amines, that is a primary amine attached to one of these base rings, can be removed. This is it can be a spontaneous event, or it can be promoted by some chemicals. You can also get what's called depurination. So if we look at this, we have this glycosidic bond here between our sugar and this nitrogen. That can be hydrolyzed. It doesn't happen frequently, but it can happen. This is called depurination. And so these kinds of modifications can be problematic and can lead to disruption of double-stranded DNA. It can prevent or reduce the base pairing if you lose the ring. Okay. There can be these nitrous acid precursors. So sodium nitrite, sodium nitrate are common preservatives found in meats, especially things like ham and delicatessen meats, so bologna and salami and so forth. They're used extensively to preserve these, but they can be converted into these nitrosamines. Nitrosamines can be further modified to produce nitrous acid, and nitrous acid promotes the deamination of these extracyclic amines. So these can be lost, and that can be problematic. And finally, there are alkylating agents. So these again are not things that are normally part of our diet. They're not normally present in organisms, in our cells, but certain kinds of exposures to these chemicals can lead to modifications of, let's say, an amine group. You can add a methyl to it, and that causes disruption of its base pairing. Most of these damaged DNA bases can be removed. There are mechanisms that the organism has to remove these and replace these with an appropriate base to allow for base pairing. So, okay, questions? Most of this that you need to know is well summarized in the book and sufficiently so on the slides. So this is mostly introductory. Remember, we had talked about amino acids and proteins. Then we talked about this other large category of biomolecules, carbohydrates. We are talking about nucleic acids. We'll talk about another one in a day or two called lipids. And that will pretty much introduce us to the basic biomolecules that we encounter in nature. Let me move this story just a little bit further forward. I had mentioned that I had found a National Guard unit and had joined it just as I was graduating from college or finishing college. I went off to basic training. I had wanted to go to church, but that first Sunday I was in this kind of holding unit without really much structure to it. nobody to ask, no way to know if there was the possibility of going to church. So that Sunday came and went. I then was assigned to a basic training unit with very clear structure. And so I happened to notice that there was a printed schedule of meetings on bases, on this at Fort Jackson. And one of them was the Church of Jesus Christ of Latter-day Saints had a weekly meeting at a particular hour in a particular building. And so I made note of that. I got up my courage and I approached my platoon sergeant to ask him if it might be possible to go to church on Sunday. And so he inquired of the company commander, and I was given a base pass. And on Sunday, I was going to be allowed to walk across the base and go to this building and be able to attend church. Well, that Sunday came, and I was very desirous to go to church. Basic training was not as much fun as I'd been told. And so I made my way out. It was a warm day. I went over to where I thought the building was to be located, and I couldn't find it. I couldn't find any signs of lots of people gathering to a building. I wandered around the base. I finally stopped some military police and asked them if they knew where this was. Nobody seemed to know where this building was located. And so after a couple hours of sort of being in the hot sun, I came back to base and I had missed the second Sunday going to church. Thank you very much. We'll pick it up Wednesday.